All right, welcome back to part 3 of the adaptive immune response. I really like this diagram because it kind of wraps up the entire response just in a neat little package with a bow on top. You do have this diagram in your book as well. So what we're looking at here is just a schematic that is outlining everything that's happening in the adaptive immune response. If you look at the side of the picture that is green, this is depicting the humoral adaptive immune response. So it's going to focus on antibodies and B cells. The tan side is focused on the adaptive immune response and features T cells. The primary response is shown in green, and the secondary response is shown with the blue arrows. So I'm just going to walk through this whole diagram with you just to kind of tie everything that we've been talking about all together. So we have our first exposure with the antigen here at the top of the picture. And this exposure, this antigen can kind of set off our immune response in a couple of different ways. Like, for instance, free antigen that is in our blood and in our lymph is available to directly activate B cells that have been kind of hanging out in the lymph nodes. And the B cells can act as antigen presenters to bring that antigen fragment to a helper T cell. The antigen can also be engulfed by a macrophage. Now remember that macrophages, dendritic cells, those are a part of the immune response, and so they don't require any specificity, and they are just going to engulf this antigen and display it with a self protein on the surface of their cells, and they're going to carry it to the helper T cell. And so this is a process called antigen presentation that's going to stimulate the helper T cell to do a number of different things. It's going to secrete cytokines to stimulate B cells, which will then proliferate, again, a fancy word for divide, and differentiate into plasma cells that secrete antibodies and memory B cells, which are forming that foundation for the secondary immune response, kind of preparing the body for the time when or if we encounter this same antigen again. So stimulation of helper T cells by antigen presentation also causes the cytokines that the helper T cell releases to stimulate cytotoxic T cells to proliferate or divide to form more cytotoxic T cells and memory T cells. Again, the memory T cells are forming that basis for immunity by creating this foundation that is going to make our body ready to attack this antigen should it ever enter our body again. The last way that this antigen can activate our adaptive immune response is it can infect body cells. And body cells that are infected are going to send out chemical signals that are going to call cytotoxic T cells to the area. These cytotoxic T cells are going to arrive at the area, and they are going to kill those self cells that have been infected by the antigen. So all of that is happening during the primary response. If the antigen enters our body again, here during the second exposure, it's going to stimulate memory T cells, to stimulate memory B cells. It's going to stimulate memory T cells to stimulate other memory T cells to proliferate into cytotoxic T cells. It's going to stimulate the memory B cells to proliferate into plasma cells and to secrete antibodies very quickly and in very large quantities. So the antibodies are going to work to neutralize antigens that are free floating in the blood and in the lymph, whereas the cytotoxic T cells that are produced are going to kill self cells that have been infected by the antigens. Notice that where these blue arrows are, we have no antigen presentation occurring because we don't need it. We already have our foundation. We already have our memory. This memory, the found in the memory T cells and in the memory B cells, that was all produced during the initial exposure to the antigen, when our helper T cells were presented with an antigen by a B cell or by a macrophage, by some kind of antigen presentation cell. So this is why our secondary response is so fast, because we don't need to eat up chunks of time waiting for antigen presentation to occur, waiting for the antigen to just happen upon a lymph node that contains B cells that is specific for it. So that secondary response happens extremely fast, and we don't even get sick. And that's what immunity is. It's preventing us from suffering when we get infected by an antigen for a second, third, or 10th time. And so that is, in a nutshell, the humoral immune response and the adaptive immune response. Okay, the last thing that I wanted to go over with you are different kinds of humoral immunity. So there are a number of different ways that we can get various antibodies into our system. So remember, if we're talking about humoral immunity, we have to be talking about antibodies. So here's a chart that kind of summarizes all of this. So we have two different kinds of humoral immunity: active and passive. If we are talking about active immunity, it means that somehow we are exposed to antigens. If we have naturally acquired active immunity, we are exposed to antigens like in the normal way, I suppose. So check it out. We've got this guy who's sneezing and coughing, and if the droplets that he's producing contain antigens that this woman has not been exposed to before, then her body has to run through the entire primary, the innate immune response, the primary immune response, and this, in order to establish memory. So we get our antibodies here by just being infected by an antigen, and then our immune system kind of plays out, and we have antibodies now in our system. Active artificially acquired immunity means that we've been stuck with a needle. In fact, if it's artificial active or even artificial passive, it means that there was a needle involved. In this case, since we're talking about antigens, it means that we have been injected with the antigen, and our body is now going to respond to that by making antibodies. So vaccines, for instance, which are made of dead or denatured antigens, are an example of artificially acquired active immunity. Artificial because it requires that needle or an injection, and active because we are exposing our body to an antigen. With passive humoral immunity, it's a little bit different. We are inserting antibodies from somebody else into our bodies. So not antigens, actual antibodies. If we have, when we're talking about naturally acquired passive immunity, it means that we are getting antibodies from our mother, which is basically one way that we've all gotten antibodies. When we were in utero developing, mom's antibodies crossed the placenta and entered into our blood. So those were some of the first antibodies that we even had. Another way that infants have more protection by antibodies is by being breastfed. So mother's milk has antibodies in it, and then when the baby drinks that milk, those antibodies get into their bloodstream through the digestive process. If you have artificially acquired passive immunity, it means that somebody has donated blood and their serum has been separated from the formed elements of that blood, and that serum contains antibodies. People who are immunocompromised then can get these antibody or the serum, which you can see in these bags here, can get the serum injected into their bloodstream by an IV. And so again, we're talking about needles here. So if it's artificially acquired, you're getting injected, and if it's naturally acquired, then it's exposure in a different way that doesn't require a needle injection. So those are the 4 different kinds of humoral immunity, 4 different ways that we can get antibodies into our system. And thanks for watching. This is the end of part 3 of adaptive immunity, and I will see you in class the next time. Bye.